So now let's take a look at some of the different types of hormones. There are three main classes of hormones, and then there's a related type of molecule I want to talk about as well. So we can classify hormones chemically based on the types of organic molecules that are used to put them together. So we have some hormones that are steroid-based, and that means they're in the lipid category, or that they're hydrophobic, if you want to call it that. And they include things like estrogen, testosterone, androgen. There are a few others that we might mention later on, but they all have this classic four-ring structure of carbon atoms with varying different other carbons and hydrogens, etc., oxygens that are bound to them. So the base molecule for all steroids is this four-ring molecule, which is called cholesterol. Another group of hormones are the monoamines, or sometimes called the biogenic amines, and I'll talk about that, the other name for them, in just a minute. And monoamines are all derived off of one or another of two different amino acids. So they're either made from tryptophan or they're made from tyrosine. Most of them are made from tyrosine, which is, as you remember, an amino acid you learned about in Bio 181. These typically tend to be Some are hydrophilic and some are hydrophobic. It depends on the type. For example, one example of a biogenic amine is thyroid hormone. That one tends to be hydrophobic. Another example of a biogenic amine is epinephrine, and that one tends to be more hydrophilic. So they kind of span both categories. Now, peptide hormones are those that are manufactured from a string of amino acids, and that string can be a really small string. It can be just a handful of amino acids long, such as angiotensin 2, which is only 1, 2, 3, 4, 5, 6, 7, 8 different amino acids long. Or they can be really large, like insulin, which is what you're looking at over here. Typically, peptide-based hormones are usually hydrophilic. Now, all three of these that I just discussed are what we call true hormones. And what I mean by that is that they are released into either the lymph or the blood. Usually it's the blood. They circulate around the body, and they influence some cell at a location distant from the organ that secreted them. Now, compare that with paracrines. Let me erase this out of the way. Paracrines are another class of molecules that your book talks about in this chapter with the endocrine system. And paracrines are chemical messengers like hormones are, but they are not true hormones. And the reason that they are not true hormones is they only influence neighboring cells. So rather than being released into the blood, circulate around in the body and impacting some distant organ or cell, they actually only influence nearby cells. And a good example of this is a class of compounds called the eicosanoids, and I'll talk more about them on the next slide. There's another term you may come across later on in your learning about the human body, and that is a term called autocrine. And an autocrine is like a paracrine in that it is a signal molecule, but rather than influencing neighboring cells, the word auto should lead you to understand that this is a signaling molecule that influences the cell that secreted it. So in other words, I as a cell secrete a hormone, or an autocrine, and that autocrine immediately influences the behavior of me, the one who secreted it. So this is a self-influencing molecule. Paracrines influence neighbors, and hormones influence cells throughout the body at distant locations. So let's look first at the only one of these that is not considered a true hormone, and again, that's the paracrines. And I'm going to look specifically at a class of molecules that are called the eicosanoids. I guess I didn't put that on here. And the eicosanoids are a bunch of different types of molecules that are all derived from a molecule that we derive from the phospholipid membrane, and we can use that molecule, it's a type of eicosanoid, and we can alter it. And if we use an enzyme called lipoxygenase, we can create a class of paracrines called leukotrienes, which are implicated in inflammatory events. Or we can use a different enzyme called cyclooxygenase, and we can convert those eicosanoids into either some inflammatory chemicals like prostacyclin, prostaglandin, and thromboxanes. Now, notice these two pathways all center around the conversion of a molecule called arachidonic acid. Arachidonic acid is one type of eicosanoid. As you might figure from the name, arachidonic acid was first discovered and described from inside of spiders. And it was later realized that this molecule is later altered via either the lipoxygenase enzyme or the cyclooxygenase enzyme into all these different types of inflammatory chemicals. And I gave you just a couple examples here, like prostaglandins have the ability to increase blood pressure. They also help to increase uterine contractions during labor and delivery. They help enhance clotting. Thromboxanes do that too. So the idea here, remember, with all of these paracrines is these are localized chemicals. They only act locally on neighbors, therefore we cannot consider them true hormones, but they're worth at least mentioning so you know about them. So now we can talk about the true hormones. These are the molecules, signal molecules that are released into the blood, that circulate around the body, and then influence some cell that's at a distant location. And we're going to talk about first the biogenic amines, or the monoamines. These can be either hydrophobic, or in some cases they can be slightly hydrophilic. It really just depends. Thyroid hormone, which is a good example of a biogenic amine, is one that's hydrophobic. Now these guys, if you remember from my definition earlier, these guys are all derived from an amino acid. The most common amino acid that is used to make biogenic amines is the amino acid tyrosine. And that's what we use to make things like thyroid hormone. And also to make all of our catecholamines. That's norepinephrine and epinephrine and dopamine. Okay. There is one other amino acid that can be used to make a biogenic amine, and that's tryptophan. Tryptophan is used to make serotonin, and that can then be converted into melatonin. And we'll be talking about those, particularly the melatonin molecule, later on. For now, that's all we need to worry about, is just that they're derived from these individual amino acids. We'll learn later, for example, how thyroid hormone is made. Steroids, as I mentioned previously, are all molecules that are derived or made from cholesterol. Here's my cholesterol molecule, and every single type of steroid, be it testosterone, estradiol, which is a form of estrogen, or cortisol, or aldosterone, these are all examples of steroids. Notice that they all have this same, and progesterone too. Notice that they all have this same four-ring structure. This is a very hydrophobic structure, very lipid-based. All of our sex hormones fall into this category. Corticosteroids fall into this category, and a lot of the hormones that are used for building muscle mass. Now, again, because these are hydrophobic, we have to use transporter proteins in the blood to get them from point A to point B. And also remember that they have the ability to enter directly into a cell to signal that cell. They do not have to rely on secondary messenger systems the way hydrophilic molecules do. The last major category of hormones are the peptide hormones. Peptide hormones are basically varying lengths of amino acids. They can be as short as only 3 amino acids long and be considered a peptide hormone. They can be up to a couple hundred of amino acids long. In general, they are typically hydrophilic. That means that they can be transported in the blood without having some protein attached to them, but it means they cannot pass through the plasma membrane of a cell. This route is blocked off to them, so they have to use the secondary messenger system. So here's a peptide hormone. It binds to a hormone receptor. You get the release of the G protein like we learned about previously. That activates another enzyme such as adenylate cyclase and eventually produces a secondary messenger molecule such as cyclic AMP, and then that can impact all kinds of different activities in cells. Sometimes it turns off enzymes, sometimes it turns on enzymes, sometimes it induces transcription of genes, sometimes it turns off transcription of genes. So some examples of peptide hormones include antidiuretic hormone, which helps prevent you from getting dehydrated, and oxytocin, which is the hormone that is important for inducing uterine contractions during labor and delivery. So that's all on the general characteristics of hormones. Next up, we're going to see how hormones can interact with cells and on how they interact with each other.